Thank you, Wayne, and thank you, ILSI, for the invitation. And so I guess you heard from my introduction, I'm sort of a lifetime breeder, partly in thanks to meeting Norman Borlaug. I guess a lot of people were inspired by him. I guess as part of the change after the break, we're starting in on food composition. And so what I've had an interest in recently is nutritional breeding, or breeding for food compositional changes. And the title of my talk here is a little bit different than what's in the abstracts, which has the example of common bean, and I will concentrate on mineral biofortification. So I'll define what biofortification is, and the mineral targets are iron and zinc. This is thanks to funding through all the CGRS centers from Harvest Plus, which has its motto as breeding crops for better nutrition. USAID and Danita in Denmark were the initial funders of the work. The outline is introduction, very small, basically on what biofortification is. Then some methods on food composition analysis from the perspective of a plant breeder. We have to use things that are quick and dirty and as cheap as possible. So sometimes it's not the best, but usually it works okay. Can people hear me? Then I'll go into inheritance in cultivars and inheritance, or the genes involved. And it was a wonderful introduction from Sherry to know about QTLs, because most of these traits are quantitatively inherited. They're not as invisible traits. They weren't targeted by early farmers, perhaps, except the fact that the women selectors saw their kids growing faster and taller, and so they said, oh, that must be a good bean variety. Then I'll discuss a little bit of breeding activities, some additional studies to think about in food composition from a plant physiology perspective, and conclusion. First, the introduction of micronutrient malnutrition, because I'll focus only on micronutrients, on mineral micronutrients, and it affects nearly half the world's population. So this is one of the biggest health problems in the world. It includes both iron deficiency anemia and zinc deficiency. In iron deficiency, you have approximately 3.5 billion people. It affects more women than men with a higher iron requirement. And preschool children also tend to be anemic, especially in developing countries. But it's also important in developed countries, and this is especially so in the case of zinc deficiency, because really the sources of zinc are limited in the diet. There's not a lot of fortification, and so legumes have been determined as one of the more important ways to reach people with zinc. Zinc is important in immunity and the use of other vitamins, and so it affects childhood stunting and then growth and development. And what is biofortification? It's now a trademarked term, but it's in the public domain for a lot of people. It's the use of plant improvement to increase the mineral densities in crop varieties so as to improve the micronutrient status of consumers. And it was focused mainly on major staples such as rice, wheat, maize, or common beans. So obviously vegetables play a big role on this. And this is a big program, multinational, and it's been taken up by national governments in China and India and Brazil. And the focus on staples was initially to get the high consumption rates that would be needed to deliver higher amounts of minerals. And of course, deliver higher minerals to women and their infants at an early stage, and that picture with the bicycle is really to show that minerals contribute to work efficiency and strength and stamina. What are the biofortification improvement needs for meeting RDA standards in rice and beans? Well, if you take the current amount in a normal variety of rice or normal amount of bean that is non-transgenic and that hasn't been improved by plant breeding, you need to increase by 12 to 20-fold 12 to 20-fold for an increase in the iron amount to complete the RDA necessary. With beans, because they're already naturally higher in iron, only a 2 to 3-fold increase would increase the amount of iron delivered to the recommended daily allowance. You can see that zinc is a little bit rare in the diet, so you have to increase by a larger amount. But this shows that in plant breeding terms it was doable. And in transgenic terms, you can even get an overdose of some of these minerals. The vitamins A, folate, and B6 are good in some of the legumes. So it makes importance of a diversified diet, even if it's a diet based on only staple grains instead of much meat. Then you have to think about bean consumption. And what is bean consumption? In the USA, there's very little consumption, only 3 kilos per person per year. But in most of Latin America, 14, 15 to 20 kilos per year is normal. And in parts of Africa, like Rwanda and Kenya, the bean consumption is very high and replaces even some of the cereals. Therefore, it's a good way to deliver different minerals and change food consumption substantially with one zinc product. The idea of biofortification is to reduce the prevalence of iron deficiency. And iron deficiency in a population in a typical country is below the recommended amount of 12 grams in blood of iron status and hemoglobin iron status. And so the idea of biofortification is to push that distribution towards iron sufficiency as a general characteristic of the population versus iron deficiency. And the leader of Harvest Plus is Howdy Boyce, who can give a lot of information if you look up reviews by him. What else is important in biofortification? Well, bioavailability is an issue, and bioavailability is the proportion of a consumed nutrient that is digested, absorbed, and utilized by an organism. Here it's determined by other elements of the same food and the cuisine or the diet, and also by the nutrient status of the consumer. Someone that's anemic will tend to absorb iron better than someone who is iron sufficient. There are promoter substances in meat that include the high sulfur amino acids. Vitamins can help, so drinking juice with your plate of beans is not a bad idea. And then certain lipids. There are anti-nutritional factors that turn out to be also health-promoting factors, such as phytates, polyphenolics, some of the minerals, calcium and manganese, fiber. So we don't want to play around with these too much, but they do affect how much you can absorb iron, and zinc is generally correlated with iron amounts in terms of concentrations. So here, concentrating on beans as an example. Beans are a very variable crop. It was under 2 domestications, and there are over 40,000 land races held in germplasm, 120,000 land races in germplasm banks, but 40,000 in one site in Colombia. And of these, 3,000 have been tested now for iron and zinc content. And of course, the seed coat colors are very different, and that implies that there are tannin differences, that there are differences in polyphenolics as well. But one thing that was interesting is that breeding and selection has generally just done an average job on an invisible trait. So this is the average iron content of beans, 55 parts per million of the total weight of the bean, and zinc, 35 parts per million. And these are varieties from Rwanda and Central America where iron deficiency is important, and you can see that the release varieties are average. So how do we get above that? We use genetic diversity. This genetic diversity is found in that collection of 40,000 genotypes, 30,000 germplasm entries that have been evaluated, and we call them a core collection or a reference set of varieties. And this is the range in mineral concentration in parts per million done by ICP analysis from 20 parts per million to 120. And one thing that's interesting in the case of the difference between wild and cultivated beans and domestication is that we don't see a huge difference. We see that the domestication events, which there were 2 in beans, had more of a role to play in creating large beans that tend to have larger amounts of iron and zinc, and small beans that tend to have smaller amounts of iron and zinc. But this wild collection were only indicated that there were a few genotypes with a higher amount of iron. And in the case of zinc, there was actually not much higher amount. The Andean and the Mesoamerican are the two domesticated gene pools, and the Andean is much more limited in zinc than the Mesoamerican, whereas vice versa, the Andean tends to have a higher iron content. So I'll give some of these discussions when I discuss the genes involved in creating high iron beans. And this was a set of studies conducted from about 2003 until publications in 2012, analyzing iron and zinc content in interdomesticate recombinant inbred line populations, phytate content in the same populations, iron and zinc in wild by cultivar populations to look at what were the effects of domestication, and determining the inheritance or the genes involved in all of these populations with that terminology that Sherry introduced so well of QTLs, and which Flavio says is the future for molecular breeding. This is really selection of genes underlying these quantitative trait loci. Whenever we start these kinds of populations, we can use large sets of genotypes like association mapping panels, or we can use recombinant inbred line populations. The interesting thing about this is that it's a discrete amount of diversity with two typical parents that diverge in characteristics such as a high iron plant crossed with a low iron plant, and like in Mendelian genetics, we determine the inheritance and the number of genes that might be involved. So we used 3 of these types of crosses. Some were from the large-seeded Andean, some were from the Andean large-seeded by small-seeded, and we analyzed them for either iron, zinc, phosphorus, phytic acid, or just phytate and phosphorus. We used various field sites, and we used a method of QTL detection, which I won't go into detail with, just to show that you need at least 2 sites whenever you're doing these trials, because food composition varies based on the place that the food is grown. The analytical methods might be more interesting to this audience, and that's iron and zinc content, which was analyzed in a quick and dirty way. And we started with coffee mills, and of course coffee mills are made of iron parts, so we got iron contamination. So we moved to aluminum as a grinding method and zirconium as the balls that actually do the grinding of the grain. We then moved from aluminum to Teflon chambers because we discovered that aluminum was a good value for soil contamination, because soil is predominantly either aluminum or iron. So if you get, especially in the tropics, if you get a high aluminum, it shows you didn't clean your seed well. You need to do the analysis again. We quantified the minerals by atomic absorption spectrophotometry, which is a cheap and old method, but we also used the ICP method by a collaboration that we had in Australia, when the Australian dollar was still a good price, a good deal. And this method was ideal, but it still was costly for the number of samples that we were doing. With repetitions, with the number of genotypes, and with genetic analysis, you're always talking about 1,000 samples. So $10 means a $10,000 study, just for a few genes. So we wanted to be as efficient as possible. We were able to analyze phytates a little bit more easily, and this was with the Wade reagent, which actually measures iron absorption to phytates and can be quantified colorimetrically. And then this was confirmed to be very accurate with HPLC quantification, and most phytates in beans are IP6. Okay? There are very few of the lower isomers, and the lower isomers would be more useful but are very difficult to study, and that would require HPLC analysis for every sample. The quantification of total seed phosphorus is also a proxy for phosphorus, and this has been done in soils for a long time. So just using molybdenum blue and atomic absorption spectrophotometry, you get the information very quickly. just to show that it's a quantitative trait for the minerals, we could look at one population for iron in 2 locations, here and here, and zinc concentration in two locations, and we see that there's always a normally distributed curve, which means that this is a quantitative trait that's controlled by many genes, which you would expect, because iron needs of the plant are multiple, both during growth and development, during photosynthesis, and for establishment of the germinating seed. So all of these steps lead up to how much iron or zinc actually gets into the bean seed itself. But we did find some QTL, and these are graphs like Sherry showed, where a chromosome is shown at the base. Each one is a different chromosome, each little graph, and wherever it surpasses a threshold line, we can declare a statistical significance, and in many cases there was a high statistical significance for iron. And many of these QTL were on one chromosome. For zinc, that previous slide was for iron. For zinc, we only found a few somewhat significant QTLs, and they were each on different chromosomes. So when we put this all together, we have a map of the genome of common bean with 11 chromosomes, and we can give general areas where we believe that among the 30,000 genes of the typical plant species, we have 100 here that might be important for zinc. On chromosome B3, we have 2 other parts of these 2 first chromosomes that are involved with zinc. We have 1 isolated for iron on B4, and then we have on B7 iron and zinc around a locus that is a domestication locus that was important for seed size. So there's nothing we can do about seed size because people like to eat big beans, and I'll show you very small beans in a second, but no one would eat the old teosinte-like beans that early Mexican farmers originally used. Apparently they popped, so they were fairly good as pop beans, but apart from that, they weren't very edible as your regular chili. And then on this chromosome B11, we find an interesting amount of genes that are involved, and this may indicate that there's several genes all stacked together in this region for both iron and zinc, iron shown in the red and zinc shown in the purple. So the summary was basically what I described there. When we talk about QTL, the number of genes, we can say there are 26 of them. But 5 for iron and 6 for zinc were in the upper half of linkage group B11, chromosome 11, and it explained 50% of the phenotypic variance, which is pretty good. So there's a lot of breeding that we can do with this actual QTL. Other QTL were identified in other linkage groups. How am I doing for time? Okay, so I'll go through this very quickly. I think I'll skip population distribution for phosphorus and phytate just to show that it's quantitative again in another population grown in 2 sites. And the overall take-home story was that QTLs, or genes for seed phytate content and seed phytate amount, this is based on a per seed level as well as a per gram level, have different locations, and many of them have to do with seed weight or with efficiency in phosphorus uptake from the soil. And since phosphorus is a rare element and getting rarer by the day, some people say we've reached peak phosphorus faster than we'll reach peak oil, you will be seeing this as important soon, this lack of phytates, in as much as phytates are a good thing. Sometimes they're a negative for iron absorption. The summary was a number of QTLs again, and the importance of the phaseolin locus, which was a domestication locus. In a third population, we found even more QTLs, but stacked up one on top of each other, and our take-home result was that there were consistent QTL for phytate and iron and zinc with previous studies, located on linkage groups B2 and this B11 that I emphasized before, one chromosome. So now let me show what happens when you cross a wild and a cultivated bean. This is a chili bean that you would like to eat. This is a wild bean that you probably wouldn't want to consume. It has a lot of nasty hemagglutinin and lectins and problems that would give you a tummy ache, and it's very small seeded, only 8 grams per 100 seed. A typical bean, and the most appreciated beans in the world, reach 1 gram per seed, or 100 grams per 100 seed. These are the favela types in Spain. They'll pay 10 euros a pound for it. And the one big difference that we should start to look at in physiological terms is wild beans have thick seed coat relative to the embryo that's inside, whereas the cultivated beans have relatively thin seed coat, but they start to have many colors. So these affect iron absorption, and this is an area for greater study. One genetic way to study is to cross these two beans, do a big field trial, and try to detect QTL. What we found when we did the QTL study was, first of all, variability for seed color, and quantitative inheritance again, always with the wild being very high in iron or very high in zinc. We found genome coverage and wild introgression, and we found QTL, but these QTL were actually different than the QTL found in the cultivated types. That chromosome 11, that chromosome 2, were not represented as well. Here we had a chromosome B5 and the same domestication gene on B7 and perhaps a seed coat color gene on B8. So all of these things, it's very important for geneticists to know all the background of his or her crop to be able to determine these new food compositional traits and what they might be associated with. So that's why I show this slide again. All of these different colors and all of these different seed sizes, which are called cultivated races, and how they're derived from these wild beans can help determine how to best biofortify bean. Of course, breeding can go forward, and breeding involves the challenge of incorporating higher mineral content, lower phytate concentration into superior commercial and agronomic traits. And these must be attractive to farmers so that they're adopted. We did one exercise of taking a low iron recurrent parent and using a small brown seed to cross by backcrossing with this genotype and obtaining new varieties that have already been released in 5 countries among these two different lines, one being determinate and one being indeterminate. And you can see the seed color is acceptable, so it really hasn't changed all that much. The additional studies that need to be done are the effect of soaking, because beans are soaked before eating, and you get a lot of leachate, and some of that leachate contains iron and these polyphenols and tannins. Here we show one variety, that variety that we used for improvement of the iron content, being a very low leaching variety. It leaches only 3% of its iron, and it plumps up very well. So in fact, it's a very good bean to have started our biofortification work with. The other important item is the location of seed iron in common bean genotypes. And here we see the embryonary axis, the cotyledons, and then the seed coat, which is the only maternal tissue. And here it is in light microscopy. And you can see here that dividing up by the embryo, the seed coat, some genotypes have a lot of iron in the seed coat from the maternal tissue. And so that's very important, because if those seeds are dark colored, much of that iron won't be available. So we really have to think about where the iron is, especially whether it's in the embryo or this cotyledon that seems to accumulate a lot of iron. I'll skip this and just show some pretty pictures with Prussian blue staining, where you can do a quick and dirty assay of what's in the seed coat, though the colored seed coat makes you not able to see the blue iron, and then the blue staining of a low iron variety, an Andean with a small amount more, and then our improved variety, which is dark bluer, okay? And this dark blue color really indicates that we were successful at making a biofortified variety. If we look at the tissue level location in the actual cotyledons within the seed, you can see that it's concentrated in these little vascular bundles. So that's another thing that we need to look at in terms of the seed not being all the same, but having this placement of iron. And this varies according to high iron, low iron. This can be confirmed by PIXI analysis, which looks at elemental iron. And again, you see the bundles with the amount of iron concentrated there, and then this strange layer around the edge, which we still don't quite understand why it has a smaller amount of iron. This is a scale for iron. So they're very high concentrations in these bundles, which is why they stay blue. And then finally, genotype by environment effects are minor. This just shows our high iron variety being bluer under a greenhouse field and under 2 years. So the conclusions are the inheritance of seed iron and zinc and phytates are predominantly quantitative. The genes are independent for iron and zinc in some case, and from the phytates, which will allow us to breed in one direction for minerals and the other direction for phytates. Localization within the seed is important, and perhaps is important for other nutritional characteristics too. And then for the next era of breeding, the most efficient markers are needed to select QTLs for high micronutrient content. I'll skip over the future steps, but it's obviously breeding for this on a larger scale with more varieties and perhaps with more legumes. So with that, thanks a lot, and eat beans. The two questions were first, whether a transgenic approach has been contemplated for biofortification in beans, and as a difference to mineral biofortification in rice, where ferritin has been used with some success, but not at a commercial level at all. Beans, there's no idea of doing it, partly because of the difficulties in transforming beans. Then the other question was about low phytate mutants, and that is a good approach. There's been some attempts in many legumes for this, and beans as well.
Hi, Owen Tuckinga from USDA. Two questions. When you're working with a wild by cultivated cross, what fraction of the population you made, your eye population, would be unpalatable due to the lectins and hemagglutinins? And then the second related is, how long would it take to bring something to market? How long would it take you to develop a new cultivar with such a schema?
So I skipped over that pretty quickly. It was called the advanced backcross method, which was pioneered by Steve Tanksley, and it's to use the wild species. In this case, our wild accessions, Owen, were the same species as Phaseolus vulgaris. They're just grown in the wild. They cross freely, and so I think in the U.S. there would be no regulatory problems with actually releasing it. We actually didn't make the graduates eat the beans, so we don't know whether there were any adverse effects in lectin and hemagglutinins, but we did try to do a double backcross, so 2 sets of backcrosses, and we did this purposely so that we would get back to type. We were surprised how fast we got back to type. And we also used a Mexican bean and crossed it into a Colombian commercial type, and it actually gave some yield advantages too because of the extra diversity. So there might be a release of one such wild by cultivated bean in Michigan, in a black bean. Correct. So repeating the question, whether the soil iron and zinc levels affect grain levels accumulation, and basically that is an issue for genotype by environment interaction. And what we find is that most tropical soils do not have iron deficiency, but zinc deficiency is very common. So we find more of an influence with zinc deficiency. But I know for the people that work on soybean adaptation to iron deficient soils, that it's very important for them to look at uptake mechanisms. We don't look that much at uptake mechanisms in iron, but in zinc we are very interested in what could be the mechanisms. Also zinc, people are recommending a foliar application, and that's been a very successful part of biofortification in wheat and some cereals.